Hi everyone, Kevin Davis with Julianna LeMieux here in New Orleans, the Big Easy at ASGCT 2025. We're in the second or third day. One loses track of these things. Julianna, how are you finding the show so far?
Good. Yeah, I just got in yesterday, so first full day for me, but it's been great. I mean, ASGCT is always pretty remarkable. The science that's going on, the work that's being presented, both I was in talks this morning, just basic research on large DNA insertions to clinical trial data, in the same session,
Yeah.
actually. So that's remarkable,
Yeah.
really.
What's your sense of the overall vibe, attendance? Because you previewed the show with
Yep.
David Barrett and the current president of the
Yeah.
society just a few days ago.
Yeah. Yeah, they were saying a couple of interesting things. One was that they had the most abstract submissions to date this year, so that was great. They said attendance was reaching at that was a couple of weeks ago, but it was reaching to roughly 8,000. So yeah, it seems like
So kind of on par with
yes,
last year.
but I think like growing, and the expo floor is certainly huge. I think that's really getting bigger every year.
I think it's a shame they put the posters in a different room, which seems to sort of suck some of the attendance out of the exhibitors who are just thirsting for human physical contact. And I've heard anecdotally from a few people that a number of international students have been unable, or perhaps just chose at the last minute not to attend, rather than risk any potential problems either getting in or leaving the country at the end of the conference, which is a great shame.
It's too sad that, that's upsetting.
Yeah. But what has stood out for you so far?
Well, this morning I was in talks all morning. So early morning, 8 o'clock session was that one I was talking about, which was some basic science on long DNA insertion. And then just wrote down a couple notes here because the last story of the session really blew me away. And he noted that they had presented some of this last year, some of the, as the clinical trial was getting started.
Yeah.
But it was a gene therapy trial for ornithine transcarbamylase deficiency. So OTCD. Yep. And so he was presenting clinical trial data from the first patient dosed. And the research that really is involved here is from Jim Wilson's group at UPenn. And also they use the ARCUS nuclease from Precision Biosystems. But these are data on the first infant to receive an in vivo liver-directed gene insertion treatment. So really remarkable. And all the data were really pointing in the right direction. He's doing well, and it's really, really promising data. And the speaker was saying that, and I should say that the company that's leading this is named eCure.
Yeah.
And he was saying that he thinks that, going forward, this could really be a platform that could be used for other metabolic diseases. So it was those are the kind of talks here at ASGCT that you just kind of get goosebumps. This is really making huge inroads.
We're also enamored with CRISPR and base and prime editing and now integrases and transposases, but ARCUS is its own little proprietary system that perhaps we need to, based on these data, we should probably pay more attention to.
Well, and the excitement about long DNA insertion in general, I think, is really good. So,
right.
Yeah.
Right.
And then also we were at the presidential symposium after that one. So kicked off by Drew Weissman, of course, Nobel Prize winner. I mean, how lucky to just, when do you get to just sit back and listen to the story, basically, which he presented so nicely about his Nobel Prize winning work of the mRNA vaccine, of course, with Katie Carrico. But then transitioned into really what he's doing now, which is really exciting science. And then third part, transitioned into a lot of work. I had no idea, Kevin. Did you have any idea he was doing this? This work on really bringing vaccine technology, mRNA technology to the entire globe. And so setting up, he was saying that he's worked with the WHO, he's worked with different international groups. And the big sticking point, as I understood it, was the manufacturing, right? And so now he worked with the WHO to set up, GMP grade facilities to make the LNPs, to make the mRNA to go into the vaccines. They're starting to exist all over the world. And then he was able to convince UPenn to license all of the IP from the Nobel Prize to these facilities. And who was it he spoke about? It was in Singapore, right?
Thailand.
Oh, in Thailand. Okay, so he works with a group over there, and they have made 10 mRNA vaccines. So anyway, he's doing all this work. in his spare time.
I know. Sort of expected a Nobel Prize victory lap, and instead we got a whole ton of
to not just do totally new science about he was working on T and B cells and working on autoimmune treatments, but also bringing this technology to really a global scale. I was blown away. I did not anticipate that talk at all.
Yeah.
Yeah.
And in a related theme, in the same session, we heard from Mick McClune from the Gates Foundation talking about their not decades, but they got into it more recently. But we sort of, HIV is sort of, we think of it as out of sight, out of mind. We've cured it, right? It's pretty much a solved problem. But of course, it's been in the headlines more recently with the government cutbacks in funding, particularly to USAID. And McClune really highlighted what a detrimental impact that is going to have. And despite the fact that we have this reverse transcriptase triple inhibitor cocktail that works if you remember to take it every day for the rest of your life, for most patients, that's a very high bar. It's very difficult to sustain that.
And also, he was saying, even if you do, you still have this low level of viremia that's, leads to this like chronic inflammation. So even if you do, he says that HIV patients who do take it every day still suffer. And so there has to be a new road forward.
So he talked about a very promising work in a public-private partnership that the Gates Foundation has been funding, and that's also spilled over into some early pilots with sickle cell disease as well. So that was good. Yesterday, in one of the early sessions, I went to a session on off-target effects in CRISPR, and the first or one of the speakers in that session was Kiran Musunuru, who's going to be popping up many times during the course of this Congress. He's giving one of the keynotes on Thursday morning, and by all accounts, that's going to be a very important, groundbreaking, newsworthy talk. So we'll be reporting on that in our second video in this series. But he gave a lovely talk about the considerations that one has to make in designing gene editing experiments, the different types of off-targets, activating cancer genes, potentially meddling with coding regions, and so on. And went through some real-world examples of genes or cases or diseases where asking the audience to poll, would you go ahead with this based on what we know? Casgevy came up, which is an interesting one because we know of an off-target site on the same chromosome that could, in a very fraction or percentage of cases, lead to a chromosomal inversion. So interesting stories there. And the moderator, or the host of that session, was Claire Cleland from UCSF, who really blew my mind when she pointed out that, yes, all this progress in somatic gene editing and gene therapy, as vectors that deliver to a whole slew of tissues, not just the liver and a couple of other ones, start to really come on board and get more efficient, we're going to have to start thinking about the spillover to germline editing, inadvertent germline editing, which isn't the number one problem now, but I thought that's an interesting story that we're going to have to watch in the years ahead. What else has caught your attention, if anything?
I have a list of things I think I'm preparing now for the afternoon, for the morning. Just lots of other great talks coming up, and excited to go see some of the technologies on the floor too.
So that just about wraps it up for day one, or the first dispatch from the Big Easy New Orleans Conference Center here at ASGCT 2025. Julianna, it's been fun working with you.
Thanks, Kevin.
Look forward to seeing you for day two. We're going to record that maybe Thursday afternoon or Friday morning.
Yes, whenever we get a chance.
See it soon.
Okay, thanks. Bye.
Bye.